Hello everyone. So today I'm doing a presentation, the last presentation, on the important biochemical pathways that are needed in order to create DNA. So creating DNA does require nucleotides, and we're going to go through the steps going from glucose and carbon dioxide to create those nucleotides. So to start things off, we'll begin with glucose. So glucose can go through the glycolysis pathway, shown here. And as glucose transforms, going through different intermediates into forming pyruvate, we do create some NADHs and some ATP. At the same time, we are using ATP at the beginning. But as we proceed from pyruvate down into the oxidation of pyruvate step, we are using the pyruvate dehydrogenase complex in order to create what is acetyl-CoA. So acetyl-CoA does, and I'm a little jumping ahead, but we do create some NADHs in here, which is important later on. But acetyl-CoA is important for the citric acid cycle, right here. So this cycle cycles through acetyl-CoA, which is a 2-carbon, and brings together oxaloacetate, which is a 4-carbon, to make a 6-carbon unit. And as it goes through this cycle, it comes back down to form a 4-carbon. So as those carbon-carbon bonds are broken down, we do create some ATP, as well as some electron carriers, which are very important. So this would be the formation of NADHs here, here, and here, as well as FADH2s. So if we kind of refer back and look at that fact that there's one glucose molecule, it oxidizes down into pyruvate, which would be 2 pyruvate molecules. We are going through the Krebs cycle twice, so we're making 6 NADHs as well as 2 FADH2s and 2 ATPs from this. So, okay, so we did make those NADHs as well as FADH2s, which are important for channeling through the electron transport chain. So what this does for us is creates an important proton gradient, in which most of the protons are on this side up here within the mitochondria, and via the FADH2s and NADHs going through this complex that transports electrons and pushes those protons up. So this proton gradient provides potential energy for the formation of what is ATP. ATP that we get through ATP synthase. So we do experience some conformational changes as we have H pluses traveling down through this ATP synthase channel, in which this ATP enzyme rotates in order to take in ADP and phosphate and to create and release ATP. So as we've already shown, we've gone through glycolysis all the way down to the formation of ATP molecules, which are important as a nucleotide for the synthesis of DNA. However, ATP is also important for the energy that we need in order to proceed into some of the other steps that we need in order to make nucleotides. So other building blocks that we do need for nucleotide synthesis is PRPP, or 5-phosphoribosyl 1 pyrophosphate. So that's what we get from going through the pentose phosphate pathway shown here. We begin with the glucose 6-phosphate, which we get from phosphorylating a glucose, and as it proceeds forward, we do, most importantly, get PRPP formation, but we also do get NADPHs, which is important for some of the anabolic reactions that we need throughout our body. But as we proceed from PRPP forward, we go through some of these important steps right here. Right here, in order to make some of these nucleotides. So beginning with, for example, carbon dioxide, we take that carbon dioxide and we make a carbamoyl phosphate. And as we proceed further, we are able to create an orotate, and that orotate can then react with PRPP in order to make essentially uridylate, which is UMP. So UMP, shown here, acts as the precursor to the important nucleotides that we get, which would be UTP, CTP, and TTP. So this goes through obviously some important steps that also does need ATP in order to proceed forward. So we are now seeing the use of ATP not only for DNA synthesis, but also the formation of nucleotides with other base pairs in order to create the DNA strands that we need. So, and then if we follow back, we are still missing some important nucleotides. And so if we look at this portion here, beginning with PRPP once again, we are able to go through a pathway that essentially results in the formation of inosinate. Inosinate. So inosinate is a precursor to the formation of two other important nucleotides, which ultimately would be ATP once again, so we see a regeneration of that, as well as GTP. So here we have then the formation of these 5 different nucleotides, 4 of which are important for DNA. So ATP, GTP, CTP, and TTP, which do need to be in the deoxyribose state, not in the ribose state. So once it's in those deoxyribose states, it can be used feasibly in the formation of DNA synthesis with other enzymes such as DNA polymerase.